A recent study on making skin cells younger has recently been successfully developed. This method is believed to be able to regenerate skin cells to be 30 years younger. This research began 15 years ago when Shinya Yamanaka, a Nobel Prize winner, discovered a method called the Yamanaka Factor. According to Yamanaka and Shoot, they succeeded in stopping the aging of cells and even making them younger. They achieved this by activating 4 component factors in DNA which came to be known as the Yamanaka Factor. In essence, the scientists earlier tinkered with and reprogrammed the DNA of skin cells to make them look younger. In other words, this method has the potential to transform mature cells back to an embryonic state. Researchers in this field think that the results of developing further research on this discovery are considered very promising in the future, not only for skin rejuvenation. Of course, this is expected to be able to repair damaged organs. For example, repairing brain cells damaged by a stroke or treating wounds that are difficult to heal. However, in the process of further development, the results were not as perfect as expected. The researchers considered that the Yamanaka factor still had a weakness, namely the age of the cells it produced became very young, almost like embryonic cells. Where these embryonic cells are known to be unable to produce collagen, unable to transmit electrical nerve signals, or perform other tasks. But the good news, a group of researchers led by Rake Wolf from the Babraham Institute managed to find a solution to this problem. Instead of making cells age 0, they were able to rewind the cell's age to 30 years younger. What's even more interesting is that they can induce cells to be reprogrammed in a time-controlled way, which previous researchers couldn't control for the Yamanaka factor with much certainty. This is of course a breath of fresh air and of course new hope for the medical world. Researchers are becoming increasingly optimistic about this technology in the future. In their research, Rake and his team collected skin fibroblast cells from middle-aged adults, ages 38 to 53 years. These cells they consider very important for wound healing. With age, fibroblast cells lose some of their ability to heal wounds. Using a viral vector, the research team injected Yamanaka factor into the cells. In previous studies, it took 50 days for the Yamanaka factor to restore cell age to 0. On days 10 and 17, Rake and colleagues in this new investigation chose to halt the Yamanaka factor's activity. The research team used a molecular aging clock to determine the biological age of the cells they analyzed after the Yamanaka factor's function was interrupted at these 2 points. Throughout the experiment, they watched for DNA and epigenetic changes that lead to cancer. Also oversees the creation of collagen and cell motility. The research team discovered that the 13th day is the best moment to halt the Yamanaka factor's action after going through a number of procedures. On day 13, cells are still in a young state, but they are still able to make collagen, and they are moving more quickly to the area of the skin that is damaged. Ines Milagre, a researcher at the Gulbenkian Institute of Science, said, "Understanding that we can rejuvenate cells is an extraordinary thing." This discovery, in Milagre's opinion, is a crucial first step. This result demonstrates that the Yamanaka factor can be applied with the appropriate modifications. Milagre warns that it can be some time before the method is made accessible in clinics. The Yamanaka factor's potential to induce cancer is the explanation behind this. It is also unknown at this time if this procedure can successfully function in other cell types. There are still many unknowns, Milagre said. Rake stated the same thing as well. In the future, Rake hopes to develop a better technique to activate the Yamanaka factor that won't lead to an increase in cancer risk or other negative effects. He said if it is discovered, we may refer to it as a rejuvenation factor, and it will provide a safer approach of regenerating cells.